I want to thank Patrick Marcelin and his team for inviting me to give a talk on sustainability in terms of COVID-19. And I'm going to focus, of course, on therapeutics, which is my area of expertise. Again, thank you very much for the invitation. I think you're all aware how to control at home and in the workplace this virus. Wearing gloves, washing your hands, wearing masks, keeping your distance. That's the easy part. But of course, we need things a lot better. We need prevention and treatment. And clearly, repurposed drugs are going to play a big role, and they are playing a big role right now, having new drugs, new immunomodulators, antibodies, and vaccine. But also hand in hand with treatment and prevention, we need testing, and clearly getting effective and robust methods of testing people to find out if they're infected, whether it's PCR-based or antigen-based or even serology-based, is quite important. And that goes hand in hand with treatment and prevention. The clear rationale for antiviral therapy is that it will be complementary to any vaccine that's being developed. There are many people who will not take the vaccine or for various reasons. And there are also the people who do not mount a vaccination. It's already happening. We need drugs for obvious reasons, like influenza. We have vaccines, but we still need Tamiflu and Xofluza to be able to tackle this virus and prevent the full-blown disease. So antiviral agent can be used to prevent progression to disease in those exposed or to treat an infection if the disease occurs in outpatients. We also need pan-coronavirus coverage. And the compounds that we are developing today have to have these type of properties. It's going to be very important because this virus is evolving, as you've heard. So we anticipate viral drift and zoonotic transmission, which is already happening. And I think it's clear that this virus, any ACE2 receptor that is available to it, it will bind very, very swiftly and very tightly, and the virus will be able to enter the cells and cause the havoc that it does. So clearly we need to prevent that as well. Right today, what we have is what I call the poor man's approach to this drug discovery. Basically drugs that are low cost and don't take too long to develop because they're already in phase 2 if they're already being used for other purposes. So this makes it very simple. The FDA approved drugs already have defined safety profiles and selected doses, so we don't have to reinvent things. There is also a lot of high throughput screening for established libraries, but that involves luck, no rational design, no rational development. And then we have drugs like nucleosides. Of course, there is a rational selection criteria because we know what the target is, which is usually the polymerase of the virus, and hopefully there's selectivity vis-a-vis the cellular polymerase. In terms of categories of compounds that are currently being developed and targeted for SARS-CoV-2, there are 5 different types. One is the anti-malaria drugs. There are the monoclonal antibodies, which have turned out to be quite effective, especially if they're used early during treatment. There are at least 3 compounds that are in the clinic that are RNA-dependent RNA polymerase inhibitors, or they cause some form of mutagenesis. There are the protease inhibitors, which are quite powerful, and they also have activity on other RNA viruses, not just COVID-2, so they make them broad spectrum, but they have their limitations. And of course, for late stage disease, where you have the cytokine storm, anti-inflammatory agents and steroids are becoming more and more important. But hopefully one day we'll be able to avoid all this by having drugs that work early on the virus and prevent the progression of disease. So the 3 drugs that are currently being developed, the nucleosides are being developed in the clinic, are remdesivir, which is already approved by the FDA, is the first one, and clearly it does have an effect in terms of reducing the hospitalization date, but also it's limited in terms of intravenous delivery. We have 2 other oral drugs. One is an old drug, NHC, what we call NHC, N-hydroxycytidine. actually originally discovered at Pharmasset. This compound in the isopropyl form is quite effective. It's a prodrug that is absorbed orally, and it works very well in terms of potency in cell culture, as you can see from the table below, but unfortunately it's known to be a mutagen. So in other words, it will have to be given to older patients who are not in the reproductive age, and also monitored very carefully. And AT527 is a drug that's being developed by Roche in collaboration with an American company. And this particular drug, the company is called Atea. It's the triphosphate form of the active metabolite is the same one as formed by PSI 938. And those of you familiar with the HCV field will remember this is a Pharmasset compound that unfortunately didn't make it because it had the caused liver abnormalities. But nevertheless, if you give it for a short period of time, you may not get these side effects. We've also been developing nucleosides. We haven't remained idle. We have a compound called 1312 that looks very good, as you can see from the table below. It works not only in viral cells, but in CALO3 cells. It's certainly as potent, if not more potent than remdesivir and some of the other compounds mentioned there. And it's also very, very non-toxic. So we're looking for safety, especially for outpatient use. That's what we hope to develop 1312 for. We also have another class of compound that could be combined with the nucleosides or the protease inhibitors. We like protease inhibitors because they are usually pan-corona active, and they have high therapeutic indices since there's no known human AMPRO orthologs. And these compounds have a limitation from the point of view that there's drug-drug interaction potential, and also they can be degraded fast by CYP enzyme, but clearly we can modify these molecules to be more stable and reduce the elimination of these drugs from the body before they have their impact on the virus. You can see from the table below, we have at least two drugs down there that are quite potent coming from our lab, and we're developing these compounds now. They're being tested in animal models now, and hopefully we'll see them in the clinic soon. There are many anti-inflammatory agents that are being considered, including baricitinib, which is now approved. Ruxolitinib, they found out is not as good as baricitinib. The trial was conducted by Incyte and Novartis, but unfortunately it didn't show any benefit. Baricitinib drug that was studied by NIH and by Lilly turned out to be quite effective, probably as effective as remdesivir, if not more. We'll see later on in a couple of weeks' time how it does against placebo and how it does versus dexamethasone. Methotrexate is also a very interesting old drug that's being recycled for COVID. It has its own interesting properties, and data is coming out showing its anti-inflammatory effect. So there's going to be a lot of interest on this molecule because it's ready off the shelf and it's widely used by a lot of people. One of the limitations is going to be the emergence of variants, which is already happening. This is a partial list of the various lineages that are emerging all over the world, from the UK to South Africa to Japan to Brazil, and now into the United States. The latest one being from the US with the 427, 1.427 and 429, sorry. These are viruses that are emerging and quite resistant to a lot of the vaccines that are currently available. So we're quite concerned about that, how much of an impact these variants make on the virus. It's likely the virus is evolving due to selective pressure and the immune suppression that's going on, and the virus is able to escape and cause problems. So we'll have to constantly be developing new vaccines. And again, that's another reason why we need to develop new drugs to counter these problems. And the reason why we need to vaccinate people as quickly as possible, as many as possible, so we can get herd immunity eventually. So resistance to vaccine is a definite. It may require novel boosters every 6 to 12 months. Resistance to therapeutic is less common. It can be done in cell culture. I don't know if it can be done in animals or, for that matter, in humans. We'll see, because we don't have many drugs yet, but we'll see what happens during the duration of treatment, usually 5 to 10 days. It's unlikely, but it's possible. We need to definitely focus more on innovative drug discovery rather than repurposing drugs. I think the designing drugs is going to be very important, and hopefully more funding will come through for innovative drug discovery, because this virus is not going away. In case you're thinking it will go away soon, it will not, even with herd immunity. And even people who are doing well now, they may have recurrences later. The discovery of drugs goes hand in hand with diagnostics. We will need to develop rapid and robust diagnostics that take into account the mutations and the variants that are occurring. So we'll have to be able to diagnose this, and always we have to also there's a lot of hearsay with the drugs that are repurposed. And it will be very important to have double-blind controlled studies for clinical studies, and also speed. And we need to develop better methods for controlled studies. We need to develop drugs for outpatients. That will be a priority, I think. When patients come into the doctor's office and they're COVID positive, what do you do with them? You don't know if they're going to progress to full-blown COVID or not. You need to give them something to prevent the progression if possible. Most patients, as you know, recover, but a lot of them don't. We know also a new phenomena is the long hauler, or the long COVID as they call them. These are patients who recover from COVID and completely recover, and then we have a recurrence and all sort of side problems like cardiovascular problems, the mental health issues, and other complications that occur with these patients. They have to be followed long-term again, and this is something we're still learning about. I believe also we may need combination chemotherapy of antiviral and anti-inflammatory, or eventually 2 antivirals together, like we do for HIV or hepatitis B, should be developed and evaluated. Clearly, that's very important. So I'd like to end this by giving acknowledgement to my group. We have a group of about 70 people, but there's a lot of people just mentioned here, as well as people at Yerkes Primate Center and my collaborators at different parts of the country, and also, of course, funding from NIH. Thank you very much.